research starts from trying to know how animals work. And with corals, the interesting things for me are, for example, calcification or the symbiotic algae they have inside, which makes them a lot of their nutrients.
By working with a coral reef experimental ecologist, a cellular physiologist, and a coral reef biogeochemist, we could really develop a project that looked at corals' response to stress at a range of scale from the molecular all the way up to the ecosystem.
The important thing would be to try to see how corals work at the physiological level and how that affects their responses to the environment and how, let's say, climate change might affect that.
Our goal is to protect coral reefs all around the world. And to really do that, we have to understand what's happening to the corals in their natural environment. By sampling the corals in these habitats with different conditions, we can see naturally what kind of changes happen. And then by moving the corals between the sites, we can see if the changes in the cells make them more like the corals that are naturally at the site, or if they end up somewhere in between. Shifting environmental conditions can change the cellular physiology of the corals. By having this whole instrument suite, we can really characterize the fine scale changes in the environmental conditions at each of the sites. Part of the challenge with a transplant experiment is to do as little damage as possible to the corals. So we have to collect the corals without damaging any of their living tissue. Then we brought the corals back to the lab where we made transplant tiles out of coral reef sediments. And then we epoxied the corals with an aquarium safe epoxy. And by doing this, almost all of our corals survived for the year of the transplant and were in pretty good condition at the end of this 1 year experiment.
If we have a combination of stressors in the wild, like warming, acidification, pollution, overfishing, then determining which of those components are more important is essential because, again, limited resources, limited amount of time. You can put a lot of time of money trying to solve one problem but is only 5% of the real problem, or you can put that same resources in something that will take care of 60% of the problem, so that's more efficient. If we had unlimited resources and limited time, that's easy, but we don't.
If we didn't understand how human anatomy worked at the cellular level, we wouldn't have many of the treatments and cures for many of the disease that humans face. And by understanding corals at the cellular level in a way similar that we understand human biology, we should be able to develop new cures and solutions for some of the diseases and other stresses impacting coral reefs. I think it's one of the most spectacular habitats on Earth, and hopefully we can bring more of habitats like this back through novel conservation efforts.